Okay, welcome back to biology. So we've spent a lot of time these past few weeks talking about what our genes are. We've talked about how genes copy themselves, how they divide in different cells, and how they are controlling those traits. But we haven't actually gotten into discussing how they actually are controlling those traits. So that's what we're doing today. How your DNA, how each gene can actually end up not just being a strand of DNA, but actually how it controls what is going on in your cell. So that is what gene expression is. How your genes, how your DNA are going to end up creating proteins that are doing or not doing things in your cell. So the flow of information in genes that is in nucleotides, right? DNA is those 4 nucleotide bases. And the DNA in a cell or in an organism is going to lead to the traits that you see in that organism by dictating the proteins that are present in that organism. So here we have like an albino raccoon. That albino raccoon is making different proteins for fur color than these more typically colored raccoons are. Here we see some albino donkeys and a normal colored donkey. So they have different genes, and those different genes are causing these to create their bodies without pigment, and this to have a body with the normal pigmentation. Okay, so proteins are the links between genotype and phenotype. So your genotype is the genes you have, and they're going to control your phenotype, which is how you look, what you can do, and your phenotype is controlled by the proteins. They look the way they look because of the proteins that their fur is expressing. They are able to eat what they eat because of the proteins that their bodies have to digest food with. Everything that they look and everything they're able to do, all of their phenotype, is controlled by proteins. Gene expression, which is the process of DNA eventually going down into protein synthesis, is going to happen in 2 stages. One is transcription and one is translation. Okay, sorry, I just got interrupted for a moment. There was a spider. So I am back now. The spider is not. So we were talking about gene expression, and gene expression happens in 2 stages, which is transcription and translation. So first let's talk about how is the fundamental relationship between genes and proteins discovered. We haven't just always, as mankind, known what genes are and how they create proteins. So let's talk about how this whole process happened. So Archibald Garrod in 1902, so a little over 100 years ago, about 125 years ago, he first suggested that genes dictate phenotypes through enzymes. So he said that there were enzymes catalyzing specific reactions, and that is how genes control your phenotypes. He noticed that the symptoms of an inherited disease that he was studying, that the patients with that disease weren't able to synthesize a certain enzyme. And so from that, he decided that cells must be synthesizing that enzyme to have normal function. And when they don't synthesize the right proteins, that's when they get a disease. And so he decided cells synthesize and degrade molecules through metabolic pathways in a series of steps. So one enzyme after another enzyme after another enzyme. One metabolic pathway we talked about a lot is aerobic respiration, right? We talked about that metabolic pathway. But that's not the only pathway. Our cells have lots of metabolic pathways to make things and to break down things, to be able to do all of the things that our bodies need to be able to do. Later, George Beadle and Edward Tatum, they had bread mold, and they took that bread mold and grew it on plates, and they exposed it to X-rays. Those X-rays created mutations in those mold cells, just like X-rays can create mutations in people in any DNA. So they created mutations in that bread, and the bread mold was not unable to survive on the media anymore. So it had lost the ability to grow. So they took, it couldn't grow on this media. They had to actually start adding these other things back to it. Their colleagues Adrian Serb and Norman Horowitz identified 3 classes of arginine deficient mutants, which each lacked a different enzyme that was necessary for synthesizing arginine. So these bread mold, when they had that X-ray shot at them, there were 3 different enzymes that got mutated in the different ones. Some of them lost one, some of them lost a second, some of them lost a third one. But any of those enzymes, when they were lost, the cells weren't able to synthesize arginine, and that's what was stopping them from growing. Because without arginine, they didn't have the amino acids necessary to build their proteins. They couldn't build anything at that point. So this experiment supported this hypothesis called one gene, one enzyme hypothesis, that your DNA is made of genes, and each gene is a discrete unit that directs the production of one specific enzyme. That is, for the most part, kind of what we still would go with. There are some exceptions to that. There are some genes that do other things, but for the most part, one gene gives you one enzyme. And if you modify that gene, if you mutate that gene, you'll have a mutated enzyme. So here's their different experiments. I don't think we actually need to go through this. It's pretty interesting if you want to, go through your textbook and pull apart what they're looking at in each of these pictures. Looking at when the mold is able to grow and when the mold is not able to grow, and the different mutations that were able to be made just through shooting X-ray at them. How it made different enzymes broken down, and how having those different enzymes that were no longer occurring, those different enzymes were broken, and so that led to a lack of growth. Okay, a couple of things that are different here. Not all proteins are enzymes. So that one gene, one enzyme hypothesis got revised to one gene, one protein. There are proteins that do other things, like make channels or do other, we've talked about a lot of functions of proteins already this semester. There's also some proteins that are made of different polypeptides. We've also talked about that when we talked about protein folding. And those proteins that are made of different polypeptides, often each polypeptide will have its own gene. So you might have several genes that give you one protein. So it's not necessarily one gene, one protein, but then you have a handful of genes, one protein. So now we kind of call it one gene, one polypeptide. So one gene leads to one polypeptide. It might take several polypeptides working together to get one protein, and your protein may or may not be an enzyme, but that's pretty much similar to that first hypothesis that they made. Commonly, gene products are called proteins and not just polypeptides, although polypeptides technically means a string of amino acids that aren't yet a functional protein, and a protein often means it is functional. That's technically the difference, but pretty much you'll just see them called proteins. Whatever that gene product is typically just going to be called a protein. In our cells, it doesn't just go right from DNA to proteins. There's a gap between them, and that is filled by RNA. So first we have transcription, where DNA is used to make RNA, and then we have translation, where RNA is used to make proteins. In transcription, we are going to be following along DNA, and it's very similar to how DNA replication occurs, only we're only going to do one gene, and it is going to be creating RNA, but we're still going to be using those base pairs to make a strand of RNA, and the specific type of RNA we make is called messenger RNA. So that messenger means that it is carrying a message from the genetic code. It is carrying an intermediate for a gene. So that's what messenger RNA. Messenger RNA has almost the same sequence as that DNA's code, and that messenger RNA is going to leave the nucleus of the cell and go out into the cytoplasm of the cell, where translation occurs. In translation, the information in mRNA is used to create a polypeptide, and that is going to happen at ribosomes. It is going to involve more types of RNA, so a bunch of different types of RNA working together, but mRNA is still carrying that message, that code from the DNA. In prokaryotes, translation and transcription can happen at the same time, so you can have DNA being transcribed, and as soon as you start to make that RNA, a ribosome can jump right on it and start making a polypeptide. So transcription can still be occurring, and translation doesn't have to wait. It can go ahead and start because it's all going on in the cytoplasm. In a eukaryotic cell, they don't ever happen at the same time. So in a eukaryotic cell, the DNA is used to make RNA for transcription. That is always going to happen in the nucleus. Then often that pre-RNA is going to be modified a bit to give our final copy of RNA. That messenger RNA is what leaves the nucleus, and then outside of the nucleus in the cytoplasm is where translation can occur at ribosomes. So in bacteria, in prokaryotes, these two processes, transcription and translation, can be linked. In eukaryotes, transcription and translation are not ever linked. With transcription is always in the nucleus, translation then always follows in the cytoplasm. Okay, the primary transcript is the initial RNA transcript from any gene prior to processing. The central dogma is a concept that cells are governed by a cellular chain of command. DNA is used to make RNA. RNA is used to make proteins. That is the central dogma. I think we've been talking about this from chapter 1. We started with this, and now we're going to end with this. This is like probably the most important concept we've talked about in class. DNA is used to make RNA, and RNA is used to make protein. This process of DNA being used to make RNA is called transcription, and RNA being used to make protein is called translation. Okay, so now that we know that DNA is making RNA and RNA is making proteins, how do all those instructions for making amino acids into proteins, how are those instructions encoded into DNA? So we know it's got all the blueprints there, but how do those blueprints work? There are 20 amino acids, but there are only 4 nucleotide bases in DNA. So somewhere between A, C, T, and G, we've got to code for 20 amino acids. So how do you know which nucleotide is going to code for an amino acid? So you couldn't just have one nucleotide code for one amino acid because there's not enough nucleotides. So if you had 2 nucleotides, that still isn't quite going to give you 20. If we have 3 nucleotides at a time, that gives us a lot more than 20. In fact, in cells, we have 3 nucleotides that are going to code for one amino acid. And those 3 nucleotides are going to be called a codon. The flow of information from gene to protein is based on a triplet code. So 3 nucleotides that don't overlap. So you have the first 3 nucleotides, then the 4th, 5th, and 6th nucleotide, then the 7th, 8th, and 9th nucleotide, 3 at a time. So here's a codon, here's a codon, here's a codon, here's a codon. This then is your DNA, double-stranded DNA, base paired. From that, we're going to undergo transcription. So make a copy of that. So if you look, this copy of DNA is almost the same as this copy of RNA. The only difference is, if you remember, DNA has thymines and RNA has uracil. So wherever we have a T, we just replace it with a U. So TGG, UGG, TTT, UUU, GGC, GGC, TCA, UCA. So any DNA strand that I gave you, I could tell you this is the coding strand, and you would know to just change your T's to U's, and that's what that amino acid sequence is going to be. There's also another way to figure out what your to figure out what your RNA sequence is going to be, and that would be to base pair with each of them. So you can say what pairs with A, that'll be U. What pairs with C, that'll be G. What pairs with C, that'll be G. What pairs with A, that'll be U. That's how your cells do it. They go through and base pair, right? But you can also take that shortcut and just turn your T's to U's. So you make that decision for yourself, which you're more comfortable with, taking the coding strand and turning the T's to U's, or taking the non-coding strand and base pairing it. But either way, you're going to come up with this RNA sequence. And then from that, you're going to go 3 nucleotides at a time, and that controls what your amino acid is. So anytime there's a UGG as the codon, tryptophan is going to be put in. Anytime there's a UUU, phenylalanine is going to be put in. I'm going to provide you with a chart that you can figure out what amino acids go where, because you can't memorize all of those codes. But we do want to be able to make a protein. You do want to be able to, if I give you DNA, you should be able to tell me what the RNA strand is. And if I give you a protein, if I give you an RNA, you should be able to give me what the protein is. And if I give you DNA, you should be able to tell me what the protein is. Any of those are fair games for questions. Okay, so the words of a gene are transcribed into complementary, non-overlapping 3 nucleotide words of RNA, which are translated into amino acids. So we have 1/3 as many amino acids as we started with nucleotides. Okay, one of the 2 strands is the template strand, which provides the template for ordering the sequence of complementary nucleotides in an RNA sequence. So the template strand is always the same strand for a given gene. Anytime, this is always going to be your template strand. And your template strand is what base pairs to give your code. I prefer to look at the coding strand and not the template strand. So I like to look at the coding strand because it has the same code, just change the T's to U's. Okay. The strand used as a template is determined by the orientation of the enzyme that transcribes the gene. So that is typically going to be controlled by DNA sequences associated with the gene. So when you have a gene, you also have a lot of other stuff with it. And that other stuff is going to control how all the enzymes combined and if they're going to go from this strand or from that strand. Okay, so here's our genetic code. So I just told you I would be providing you with the genetic code when we do quizzes or tests. So if you were coming along and you wanted to know what a CUU, that's going to make leucine. If you want to know what a CAU, that's going to make histidine in your protein. So these are each of your codons. You read them by your first RNA base is on this strand. Your second RNA base is on this strand. So if you said you had A, G, C, you'd say here's A in the first one. Here's G as a second position, so those come together here. And then you can find AGC right here, and you'd know serine would go in. So let's go back. Let's go back to what your slide is saying. During translation, the RNA-based triplets are called codons, and they're read in the 5' to 3' direction. That's the way we're always moving. The non-template strand is called the coding strand because the nucleotides of the strand are identical to the codons, except T is replaced with U. T in DNA is replaced with U in RNA. Each codon specifies the amino acid to be placed in the corresponding position along the polypeptide. So anytime you have GUU in your RNA, it is always going to give you valine. It can give you nothing else. It can only give you valine. In the 1960s, all 64 codons were deciphered. Of these codons, 3 of them are stop codons. So UAA, UAG, and UGA. Those are stop codons. They're going to stop translation. So instead of putting in an amino acid, it puts in a water molecule, and that just kind of breaks the whole thing apart. You might notice also that there is some redundancy in this genetic code. So this amino acid, this codon or this codon both give you phenylalanine. All of these codons give you proline. So in one way, that's kind of nice because that gives you a little bit of wiggle room. So if your DNA gets a little bit of a mutation, you'll still be able to make the same protein. However, it's not ambiguous. So if it comes to this code, to this codon, it is not ambiguous what amino acid is going to be put on. It is always this amino acid. Each codon only gives you that one specific amino acid. There is no wiggle room for it to be different amino acids for each codon. The only wiggle room is that you have different codons that can give you the same amino acid. Codons also have to be read in the correct reading frame. So if your enzyme started an amino acid too soon or an amino acid too late, you'd have an entirely different protein made. So to make the correct protein, you have to have the right reading frame. And there will be lots of signals on RNA to tell you, how to line it up. And one of those important signals is AUG is a start codon. So every amino acid is going to or every polypeptide is going to start with that amino acid, methionine. That is always going to be the first amino acid that is incorporated. Your cell can't start a codon that's a nucleotide too soon or too late, because then it wouldn't be starting at AUG. So that is always the reading, always starts and sets the reading frame. The genetic code is nearly universal, so whether you are looking at bacteria or whether you are looking at people or plants or anything, we have almost the same genetic code in anything, to the point where you could take a firefly gene and put it into a tobacco plant, and that tobacco plant will express the firefly gene and it will light up. You could take a jellyfish gene and put it into a pig, and that pig is going to express that jellyfish gene. So we have this yellow on its snout and its feet from where that jellyfish gene is being expressed. So all types of different organisms, if you have the gene, you can make that gene product. Genes can be transcribed and translated after being transplanted from one species to another. This is important medically too, so we can do things like take insulin, the gene for insulin is a pretty common one that we do. You can take the gene for insulin, insert it into a bacteria, then you can grow that bacteria in a lab. It grows really fast, so you can get it making a whole lot of insulin. Then you purify out the insulin, and you have a treatment for people who are insulin-dependent diabetics. So that is one way that we can create drug treatments. We can create all kinds of treatments for people who are missing a gene, who are not creating a specific enzyme or protein. We can create it in a lab, and it creates the exact same protein, and we just have to then inject it or get it somehow into the person. Transcription is the first stage of gene expression, and the RNA strand is complementary to the DNA strand. I think actually I'm going to pause this at this point, and then we're going to come back and talk about transcription in the second clip. So I'll see you back in a little bit.